Thank you all for coming to our next small talk about big data. Tonight we are very pleased to have Frank Corvino here. He's a managing partner at Genesis Research, which is a healthcare consultancy. And he's going to be talking about what Genesis is doing in the area of outcomes and health effectiveness. Very interesting in that my doctoral dissertation when I was here at Stevens was focused on the healthcare area in mining safety data, and very similar to the kind of work that Frank and his organization does. So with that, I'll turn it over to Frank.
Thank you very much. Well, hello everybody. Thank you for coming. My name is Frank Corvino. I graduated Stevens in 2009 from the physics department. I got a Ph.D. in physics. And you know, with that analytic background that you get from here, it's easily transferable to many other areas. I chose the healthcare space. I represent Genesis Research, which is a company I co-founded. We are in Hoboken. We're about 6 years old. And we are a CRO, contract research organization. So as I go through this talk, which is comparative effectiveness research using real-world data, I'll give you guys a little example on what we do and where we can come into it. If at any time you have any questions, please just raise your hand and we could take them real time. I don't have a ton of slides here tonight, so I think that I can get through most of it in about 40 minutes, which should give some time for discussion afterwards. But if anything comes up at any time, please don't hesitate. Comparative effectiveness research and real-world data. These slides are also very text dense, so I'll be reading off the slides as well as you are. So first I want to define two things, real-world data and real-world evidence. Real-world is kind of a buzzword, right? Because you have these products in these pharma companies and they go through their clinical trials, which we'll get into in a bit. Clinical trials are very controlled and they're usually with a small number of patients, usually in the hundreds. What happens after these drugs launch and they're out in the public? How many people use them? Thousands, tens of thousands, hundreds of thousands. The more the better. And that's where pharmaceutical marketing strategies come into play. The thing is, now that we're seeing tens of thousands and hundreds of thousands of patients use these drugs, we kind of have a much bigger sample size. And you guys know sample sizes and statistics and how this can come into play and actually looking at the effect that these drugs are having on the population. So to define two things really quick, real-world data and real-world evidence. Real-world data is observational data collected in a real-world setting, not in RCTs. And real-world evidence is the organization of this real-world data to form a conclusion or a judgment. All right, so in general, we have real-world data, observations, gathered after a prescriptive decision is made, and the researcher has no control of the patient's management beyond that point. And a lot of times, looking at this could tell you, well, what if the data is bad? Sometimes it is. What if the data is good? Sometimes it is. How do we decipher from good and bad data? These are all important questions. Now let's look at some real-world data sources. And I will start clockwise from electronic health records. These are very popular, especially in consideration of the new healthcare laws into place. There's a lot more structure around creating EHRs in hospitals and medical care systems. Then we have our chart reviews. Does anybody know what a chart review is? Okay, I'll explain in a minute. Supplements to RCT. RCT is a randomized clinical trial, which we discussed. Registries. Does anybody know what a registry is? All right. Large sample trials, administrative data, those come from hospitals, health surveys, and claims. So before I explain what medical chart reviews and registries are, let's talk about what claims are. Claims is literally insurance claims. So I'm on a health plan, right? I go to the doctor. He diagnoses me with diabetes, which I don't have, but if I did, he'd give me a 250XX ICD-9 code and bill my insurance. So all of that data goes into a big database. And Thomson Reuters used to have one, and several third-party clients do them and sell them to us to use. So I could look at a database that could have 100 million patients in it for over 7 years from all these different insurance collected, either commercial insurance or Medicare supplemental or Medicaid, and I can see what insurance claims are being filed. This is a very, very powerful data source because not only do we see really everything that's going on with this patient, because if they have serious health conditions, they're going to want to be reimbursed and go through their insurance, but we also get to see their eligibility, which is very important when making healthcare decisions. If we don't know their eligibility, if we don't know when they're actually insured, things could be going on in these non-eligible periods that could throw our whole study off. So the claims data, US claims data, is a very, very powerful source of real-world data, and we use it a lot. Let's say there's an indication that a pharmaceutical company wants to study. And they say, Frank, go look in the claims data and give me a feasibility study. How many patients do you find? And I get back to that person, I said, I found 37 patients. He goes, that's expected. It's a rare disorder, and we're probably not going to be able to use the database. What do we do from there? Then we go to medical chart reviews and registries. A medical chart review would be, for us, Genesis, to find locations where these patients exist and pull their charts, blind their charts, and run an analysis on it. So if we can get in touch with 3 or 4 hospital systems that have a significant sample size of patients, we actually go there, pull their charts, blind their charts, and create our own database. Registries are similar, except registries are done on most products when they launch, and it's a very large scale. This is something we do as well. So company X, launches a drug. It's a biologic to treat an autoimmune disorder. There is immediately a registry done. And what that means is sites are selected where this drug will be utilized, and nurses or healthcare practitioners there will enter in the person's data. So in effect, we're creating a database, so to speak, on these patients on this drug. And we can use this for future studies. So for example, if there's an antihypertensive that did great in its clinical trials, and we want to see how it's doing when it's actually on the market, we could either do a chart review or a registry and look at this data to see the effectiveness in a real-world setting. So electronic health records are slightly different. One advantage of the claims data is that we could actually see cost information. So that opens up a whole new door of analyses that we can do. Pricing, reimbursement, value-based pricing, healthcare resource utilization costs, it's endless. Electronic health records and electronic medical records tell a slightly different story. Some of them, these holy grail type sources, have both clinical information and cost information. But most of the electronic health records only have clinical information. This is useful, but in another aspect of healthcare. With the EMRs and the EHRs, and I'll use that term, to mean the same thing, we could look at biomarkers. We could look at actual blood pressure. We could look at actual cholesterol. These are values that the claims data really doesn't have. The claims data could let us know, yeah, you have hypertension and you went to the doctor and were diagnosed on this day and so on and so forth. And we prescribed you an ACE inhibitor. But we don't know how that ACE inhibitor is working. We don't know how that's taking down your biomarkers. The EHRs and the EMRs can help with that. So that's really a big overview of some of the sources we use. And at Genesis, we mainly use the EMRs, the claims, the health surveys, medical chart reviews, and registries. I don't know if I got into health surveys. It's not a big part of our business, but it's important. A lot of times I call them patient-reported outcomes, or PROs, and they're instruments that are constructed, developed, and given to patients to reflect their quality of life or anything that they want to report. It's a patient-reported outcome. Any questions so far?
How much of this is unstructured data?
too much. A lot of the claims databases are structured, and they're structured well. A lot of the EMRs are, can I say slightly structured? Slightly structured. He's an optimist. We've dealt with some EMRs that have their gaps, and the important part is knowing the strengths and weaknesses of the source you're using. You know, primary data collection, health surveys, medical chart reviews, those are what you make of it. But, you know, some of the EMRs are still developing, and they have their gaps.
I do have a question. I guess it's a little bit technical, but how do you analyze the data? Do you use any tools like Hadoop or other open source?
That's a great question, and we're going to get to that. But mostly we use SAS, R, Excel, depending on the size.
What's the average size of a data set that you use?
Some of these claims databases can be huge. How many terabytes? 3 terabytes, 4 terabytes of data. Over 100 million patients, you know, with years and years of observations.
Is there a particular reason why are you using any type of distributed computing with SAS or?
A lot of times we have to use SAS at our client's behest because it's recognized and we can publish easy with it. Okay. You know, for quality control purposes. We use a lot of different softwares though, especially for the data visualization aspects of it, which is not really in this talk, which I would be happy to talk to you about later.
Sure.
Okay, comparative effectiveness research. And comparative effectiveness is like a buzzword these days. Everybody uses it, and it's really an umbrella term that means you're using real-world data to compare different things. It could be safety, it could be cost, it could be effectiveness. And safety, cost, effectiveness, these span over multiple departments in pharmaceutical and biotech companies. Epidemiology, marketing sciences, health economics, outcomes research, business development. We'll talk about all these. But these comparisons could be, as we have, different options of the same treatment approach. So several drugs of the same class or different classes. So you have hypertension, we're going to look at an ACE inhibitor versus an ARB, for example, and see what the pluses and minuses of each. We could look at different treatment approaches, surgery versus radiation, versus maybe nothing, versus chemotherapy, see what the pluses and minuses are. One thing to really point out here is that these approaches are often done not on the indication as a whole, but on subgroups of an indication. For example, back to hypertension, we could look at the whole hypertensive population, or we could look at stage 1 hypertension, stage 2 hypertension, obese people, elderly people, women, African Americans, or other high-risk subgroups. Because different therapies and different classes will work better or worse on different subgroups. So we could really focus this into a particular subgroup instead of looking at the indication on a whole. And then using a specific intervention and its non-use. So doing something versus doing nothing. We want to prevent blood clots. Do we do aspirin? Do we do warfarin? Do we do something else? What are the pluses and minuses with each? Or do we just watch and wait? Depending on the patient profile, we can do different things. So these are just examples of our comparative effectiveness research and different strategies. So some CER techniques. Perspective observation, data collection, meta-analyses, systematic reviews. You guys know what meta-analyses and systematic reviews are, right? One person has to. One person besides Dr. Isakowitz and Marco. All right, systematic review. There's no wrong answer. There is, but I won't tell you if it is. What do you think? It starts with an L.
Literature.
Ends in literature. So a lot of times we'll have to do a systematic review on a certain indication. And this means we take several papers, and that's really what a meta-analysis. A meta-analysis is actually taking different papers that did a particular trial and putting the results together and doing a weighted average of results with some statistical techniques that we're not going to talk about now. Systematic reviews are a little bit less statistical and more just combining literature sources. It's a systematic way to do a comprehensive literature review, which we really have to do at the beginning of every project. Database analyses, these are our bread and butter, using claims data or EMR or registry data or chart reviews. Reanalysis and inferences from RCTs. Sometimes these randomized clinical trials are so big, there's so much data, we get our primary endpoints from them, but then there's other data we can gather from it. It's still real world. Pragmatic trials. These are pretty interesting. Pragmatic trial is, let's say when the drug is out on the market, we'll do like a loose clinical trial on these patients using the real world data. What do I mean by loose? In a clinical trial, there's inclusion and exclusion criteria. A lot of times it's very strict. With a pragmatic clinical trial or a PCT, they kind of loosen up the inclusion-exclusion criteria to get more patients in and really to see how these drugs are working on a broader audience in a real-world setting. So these can go on for years, and they're very, very useful. And then we have indirect comparisons, and those are pretty much it. Big data in healthcare. All right. So now that we know what the big data sources are, what can they do for us? Well, they're very big, as we discussed. You know, some of the sources being in tens of terabytes and looking at hundreds of millions of patients. Variability, structured and unstructured. Some are nice, clean, easy to use. Others we have to clean ourselves. We've done that before with EMRs, and there's a lot of stuff I'd rather do. Complex, that just depends on the data source. Some are complex, some are simple. And we use these to identify questions to ask. A lot of this data we can use to be hypothesis generating, and a lot of this data we could use to prove hypotheses. Collect, store, analyze, interpret, and hopefully to gain new insights. No matter what new insights we gain, using big data in healthcare is never the end. It's usually just the beginning. We use this to generate questions, form some theories, prove some hypotheses, and then move on to more controlled clinical analyses.
Question, do you use any type of machine learning techniques to learn from the data?
I don't understand the question.
Like classification algorithms, support vector machines, or anything to, if you have a theory, go on with it and then discover things from the data? Or is it more of a statistical analysis saying, this person is more likely to have this disease based on an average?
Well, most of the data we have is retrospective. So I think the answer to your question is more statistical approaches. Looking at patient cohorts and seeing, who's more prone to a certain adverse event, what the treatment pathways are, mostly statistical and retroactive. Big data in healthcare. All right, I mentioned the increased use of EMR and other digital data. I think in the next 5 years, we're going to see a big surge of EMR data being used. I know with certain healthcare systems that I'm talking to now, they have great EMRs in place. EMRs that have clinical information, administrative information, and even cost and reimbursement information. The holy grail, right? But they're only 2 years old. And to go far back, then we have holes in patients' life, and we don't know where they were and what they were being treated with, and we can't really use it. But in the next 5 years, they're going to be very, very useful. Another thing is they're very low cost, respectively, right? To run a clinical trial costs tens of millions, sometimes hundreds of millions of dollars to get all this information. To do something like this, might be in the $100,000 range. So to use big data in healthcare and to guide clinical trials with it is a very, very cost-effective way of planning your RCTs, planning your PCTs, and really developing a value statement for your therapy. These can save a company millions, if not billions of dollars, just using this big data in the beginning. What else do I want to highlight from this? One thing that's not here I think I'll discuss that a little bit. There's a slide that speaks to that well.
You mentioned the new healthcare law and its impact on the availability of data. Could you say a little bit more about that?
Well, the new healthcare law requires construction of EMRs, where previously we didn't have that. So, maybe a few years ago, where we would be more dependent on actual pulling paper charts. In 5 years, I don't think there should be any of that, because now healthcare providers are required to have an EMR in place. And the reason it's going to take a couple of years is because these have to mature, and we need patient history in them. But
Those EMRs, is it a universal medical record, or is it the same for every healthcare system, or is it
it is. I don't know if it's universal, but I know that when I hear universal, I think of one stop to get the whole countries. I don't think it's going to be on that level, but I think the different plans and the different providers, the different hospital systems, are mandated to now use EMRs, which is going to make things very accessible for people looking for this data in the future. So now we could look at the advantages and disadvantages of big data in healthcare. The advantages speak for themselves, and we touched on a few of them: large populations, enhanced linkage, great longitudinal length, and I think that's key. A lot of the analyses we do using these big data sources, we could track patients for 3, 5, 6 years continuously and really look at what's happening. This is very important when looking at clinical pathways and treatment pathways of newly diagnosed patients with certain indications. If the majority of therapeutics out on the market now are not first-line therapy, they're second line and third line. We could talk about that more in a bit when we get to the treatment pathways. But having this database history and to be able to look at these patients longitudinally from when they were newly diagnosed really tells us, okay, here's what the clinical guidelines say is going to happen. Here's what we're actually seeing. Here's what these patients are doing. And in addition to looking at their treatment pathways, we could also look at adherence and compliance to these therapies. And that opens up a whole new game. So great longitudinal length and the cost are major drivers in this. Disadvantages: underreporting and error entries. We deal with this a lot. And I guess in my experience, there are certain indications in certain databases that I know are strong and representative. And then there's certain indications in certain databases that I know are underrepresented. There's no magic way. I only know it from experience. But the way to prevent a lot of these problems from happening, especially when working in a new therapeutic area, is to have 3 people on board for every analysis. One is a clinical expert in that field that could really speak to the clinical practices. The other is an epidemiologist that knows the disease epidemiology and treatment patterns, and the other is a database expert that can speak to the strengths and weaknesses of that particular data source. Putting the 3 of those people together, you could put together a statistical plan, do some feasibility analyses, and identify if this is the right source or if it's not the right source. Non-specificity, country-specific availability, that's an important one. A lot of times we do analyses in the US and EU5. Everybody wants to do analyses in Asia and South America. The only issue with this data is, we can look for data in China, but that data in China might only be reflective of a certain city or a certain socioeconomic status and not reflective of the complete population. And so we find that a lot I don't want to say developing countries, but countries that don't have good electronic medical records, we find, some issues on whether the data is actually representative of this country or a particular subset of the people who live in a certain city. And that's very important. Primary data pools of healthcare big data. We have our claims and cost data, which we use a lot and discussed. We have our pharmaceutical R&D and life sciences data. They get these through these clinical trials and screening libraries, genomics. Clinical data, our main source for that is our EMRs, and patient behavior and sentiment data. We mostly get these through primary data collection and patient-reported outcomes. So these are really the four big pools. We deal with mostly the top, but also dabble with the bottom ones as well. Pharmacovigilance and signal detection. So we should take a while and soak this one in, because this shows the timeline on the right, the cost on the left, and how we can go from 5,000 to 10,000 compounds to having one FDA-approved drug. It usually takes somewhere between 9 and 15 years from when the molecule is developed to when it actually launches. There are times we could work on these molecules for years that haven't even launched yet. Then there are times we work on molecules that are already on the market. The interesting part of how we use big data here is, let's say a pharmaceutical company is thinking about in-licensing a product, right? This is a multi-million, multi-billion-dollar deal. They have to make a decision. Where are they getting their information from? They're getting their information mainly from the person selling this drug or out-licensing this product. That person's going to tell the in-licenser that this drug is fantastic. So what they have to do is some independent research. And the basis of independent research, which we like to look at as an early economic clinical evaluation, is using this big data to say, okay, here's the drug. What are its targets, right? What are its target efficacies? What are its target safety parameters? Do we have those? Do we need those? If we have them, that's good. Then we can go into the proper claims database, or EMR, and look at the treatment landscapes, look at the eligible populations, look at how many people are suffering from this indication, their standards of care, and their treatment pathways. Knowing this, maybe then we can look through the literature, see what the effectiveness is, or look through an EMR, see what the effectiveness is. So now we know the landscape, the effectiveness, and the cost for all these marketed products. If the drug we're looking to in-license has safety parameters in this area and efficacy parameters in this area, we could throw that now into our model, which is the current standards of care that exist now, and see where it's going to be. If the target efficacies and safety parameters of the therapy that this company is looking to in-license are good with respect to the current standards of care, then it gets good reviews. If it's not good with respect to the standards of care, they might want to pass on it. Using this real-world data can really elucidate these parameters very early on. In addition, doing these analyses around phase 1 and phase 2 of the comparative landscape, not only of the therapies that exist now, but the therapies that will be on the market before you. Because remember, this is a 15-year process. So if we're looking to launch in 7 or 8 years, we can't look at what happened in the past 3 years, because 3 or 4 years from now, there will be new therapies that we didn't consider that will be on by the time we launch. So we have to take those into account too. That's not that easy, and we can't always do that with big data, because big data mostly looks backwards. This would have to look forward. But we still would have to include them in the model and, plan for it. So, any questions on this? How are we doing on time? I promised 45 minutes. I don't know if that's happening. Overview of the drug development process. So we could look at preclinical, phase 1. Phase 1 primarily looks at toxicity and the upper limits. Phase 2, we start to get into effectiveness and safety. And phase 3, and that's when we start to look at comparators and make a value statement. But even if you look at phase 3, you have 1,000 to 3,000 patient volunteers. And personally, I think that's high compared to a lot of the RCTs that I've looked at for phase 3. Normally, I'd say it's 1,000, maybe 2, a lot of times in the hundreds. But then when it gets to the real world, then we have tens of thousands, hundreds of thousands. And hopefully, as the drug companies would want, this happens within the first couple of years. So we could immediately look at these big data sources and see how it's really working. Signal detection. Let's talk about what a signal is. A signal, as we define it, is information that arises from one or multiple sources, including observations or experiments, which suggests a new, potentially causal association or a new aspect of a known association between an intervention and an event or set of related events, either adverse or beneficial. So what we're going to shift to right now is using this big data to look at signals. More specifically, safety signals. So safety signals in general depend on frequency, the type of signal, the time to onset and duration, and the presence of documented high-quality information of the adverse event. And I would almost say, not usually, but always, more than a single report is required to generate a safety signal. And big data is usually the first step. But not always. So if we have already identified a safety signal, let's look at this. So in this example, we have an oncology drug associated with a form of cardiomyopathy. Ongoing surveillance is initiated to gather more information about the association. One type of surveillance is a prospective observational study. This could be a registry. Registries are prospective, going forward. The claims database are retrospective, going backwards. And the third step, surveillance procedures, new information suggesting that the risk for this adverse effect may be especially higher in certain subgroups. As I mentioned before, not all patients respond the same way. So sometimes an adverse event is subgroup specific, and sometimes it's on the patient population as a whole. These are things we have to figure out. So signal detection. Most common sources, spontaneous reports collected from patients, providers, lawyers, health authorities. These are usually the most vague because a lot of times we don't know the denominator we're working with. And we don't know a lot of other things that are going on. But it could be a start for something to look into. FDA reports, MedWatch system, healthcare professionals and consumers, and the ACER, the Adverse Event Reporting System, which gets used a lot. You're very familiar with this. Clinical data from pharmaceutical manufacturers. This could be in an RCT, in a PCT. They saw something in a clinical setting. They're reporting it, and other sources. And here's just a quick look at drug withdrawals in the past 10 years due to detected safety signals. So now let's look at signal detection through using big data. All right, so data mining for safety signal detection. And this is the process of applying algorithms to a large data set to determine whether or not certain adverse events are being reported with greater frequency than expected. And the main goal is to identify events that were previously considered unexpected and provide guidance for further investigation. So this data mining approach is saying, okay, we have a certain amount of people with a disease, with an indication. Let's look at the different therapies, match them. This is where the sophisticated statistical algorithm comes in. And look at them over a certain period of time, from the time they started therapy to the time they leave the database, to see if anything pops up relative, pops up on the patients on therapy A relative to the patients on therapy B, or the patients on therapy A relative to the ones not on therapy A. We'll get into this in a little bit more detail in a bit. But it has three components, and one is selection. Who are we going to select? And that is patients with a certain indication on a certain therapy mainly. How are we going to score it? Most times we score it with an incidence rate and an incidence rate ratio. Does everybody know what an incidence rate is? Does anybody know what an incidence rate is? To calculate an incidence, there's incidence and there's prevalence, right? Prevalence, Dominic, what's prevalence? I got to put you on the spot. Prevalence is like, if we did a cross-section of a population, how many people have diabetes? You know, we're looking at 100,000 people, how many have diabetes? Incidence is, let's look at the non-diabetic population. How many are going to get diabetes in the next year? So we're looking at incidence. We're looking at how many people are going to get this certain thing. And that's how we're going to score it. So if this half of the room is on therapy A, and this half of the room is on therapy B, okay, and you're matched. We're going to talk about matching in a bit. So everybody is matched by age, sex, and treatment initiation, just so there's no confounding, right? For every female that's 22 years old that started therapy on July 1st, there's going to be therapy A on July 1st, there's going to be a 22-year-old female that started therapy B on July 1st. And we're going to look at everybody from the day they started treatment until they left the database. And we're going to see how many events happened in this continuous period in Group A and in Group B. What events? Any event. And that's how we're going to compare it. So that's going to be our scoring. And then comparison and detection. If the patients on therapy A had an incidence rate for a certain adverse effect, you know, stroke, heart attack, that's 2 or 3 times more likely than Group B. And the confidence intervals and statistics are high enough where we could deem this valid, then that is a potential safety signal which we're going to have to investigate further. So there's selection of patients, scoring, and comparison. All right, so here are the rules. All signals deemed worthy of investigation require further analysis in an attempt to confirm or refute the signal. 2, decision if unusual score is a signal depends on the clinical context. 3, good data is key. Knowing the data source you use says a lot to the validity of your signal. And 4, dynamiting is not enough, never enough. No matter how good the data, no matter how sophisticated the tools, it's usually a first step, and subsequent clinical analyses are necessary. Detecting safety signals through big data, more. So these quantitative data mining methods, I'll go through these really quick. Disproportionality analysis and proportional reporting ratios, reporting odds ratios and probability filtering algorithm, Bayesian confidence propagation, neural network, predictive modeling, clustering, link analysis, deviation detection, R-test, SETS test, the Q-score tests, and the chi-square test. These are all statistical methods. Signal detection, we have timing, frequency, interpreting the numbers from spontaneous report systems, and masking. These are important factors to look at when looking at data, when looking at safety signals through big data. Interpreting the numbers from spontaneous report systems, we don't really have that problem in big data. Masking, sometimes we do have that. One bad medicine making others look good, or if we have, you know, small numbers of patients and low confidence intervals, we might get, you know, crummy results, you know, based on the scoring system. So let's give an example. So here's the idea. Check for disproportionalities and diagnosis incidence rates for patients in the same indication on 2 different treatments, molecule and comparator. Confounding reduction. As I mentioned before, we're going to do a random 1-to-1 matching of patients treated on the molecule, those treated on the comparator, based on age, gender, index date, and treatment date. So index date here will be the date that you came down with your indication, right? So if we're looking, let's keep using hypertension as an example. So the day you were diagnosed with hypertension is going to be your index date. So we're going to match you not only on index date, but treatment date. So if somebody from group A had hypertension, they were diagnosed in January, we're going to make sure that match in group B was diagnosed in January. Why? Because we want to make sure that you've had the disease for the same amount of time. So matching the index date and the treatment date, as well as age and gender, are very, very important here. This really reduces confounding. Another step we could use to reduce confounding, match health state. And that's a little bit more difficult because we have our Charlson comorbidity health score that could speak to the health of a person. It's broken up into 14 categories. And we could match it by health state plus or minus 1. But as you can imagine, to get a good number of patients, after doing all this matching, we have to start with a very, very large number. And that's where the big in big data comes into it. You'll see how many we lose.
Frank, just a quick question. How do you deal with patients taking other drugs and therapies? How do you factor them into your analysis here?
That's a good question. If we have prior knowledge that patients that we're looking at in our indication could be taking drugs that also have this safety signal. So what's being asked here is whether you're in group A or B, you could be taking a drug for a different indication that has a propensity for a certain adverse event that we might not even be able to see here. And there's two answers to this. One is if we know in advance, we could exclude patients on that certain drug. Two is if we don't know in advance, when we look into this more, if we do see a safety signal, we can look at the comorbidities and co-medications of these patients showing the signal and say, this might be why. A lot of times when we do safety analyses, if we're looking, let's say, we did a recent safety analysis for a drug and looked for a specific adverse event that this drug was supposed to give rise to. So we looked at it in different subgroups, and we found that, lo and behold, there's a higher incidence of this adverse event in patients with this drug. But then once we got down to the patient level of patients who actually had this adverse event, we find out that 75% of the patients had other risk factors, either in the form of another therapy or in the form of, some health factor. Their kidneys failed a couple of days before they had this other one, and it's almost a given that when your kidneys fail, you get the specific adverse event. So by looking at it on the surface, that can, be hidden. But by getting down into the details, which is the next step after we find these safety signals, we uncover them. So here's an example. So we have some charts here. So first thing we did was pull all patients in a certain indication, regardless of treatment. We got 82,000. And then once we matched them with the molecule cohort and the non-molecule cohort, we got 20,000 in the molecule and 20,000 in the non-molecule. Obviously, it's the same number, of course, they're matched. But we also lost 40,000. And we lost those patients because, they couldn't be matched. So this would be the first step. And on the right, after the end, we could see the follow-up median interquartile range in months. And that's follow-up time from treatment index going forward. So we kind of have an idea of how long these patients are in the database for. For the molecule, it was a median of 17.8 months. And for the non-molecule, it was a median of 14 months. Right? And then we have our definitions. End of follow-up is defined as end of plan eligibility, data cutoff, end of study. Our rules to define our indication population. Our months from first indication claim to the end of eligibility. That's important. and then some rules around matching. Age plus or minus 5 years, first indication plus or minus 90, treatment initiation plus or minus a year. And then from there, we could look at our scoring. Now, what specific safety event were we looking for? None. We were looking at any possible safety event. So after treatment initiation for molecule and comparator, we looked at all their future diagnoses from that point until the end. So, for example, everybody familiar with ICD-9 coding? It's just, a medical coding. So it's what healthcare providers use as a code to describe an indication. So diabetes, for example, is 250. And it's a 5-digit code. So 250 is the first 3 digits that tell you it's diabetes. And then the last 2 digits tell you what type of diabetes. If it's 250 something 0 or 250 something 2, it's type 2 diabetes. If it's 250 something 1 or 250 something 3, it's type 1 diabetes. So that's what we're looking at. We're looking at the ICD-9 codes. So we could say, and this is with no restriction. So we're looking at the scores, but we're not restricting it to anything. So we could say, okay, in this case, 008. In the molecule, 5 people had it. In the comparator, 8 people had it. We had a relative risk, a rate ratio, which is just forming a ratio of the incidence rates of 0.7. Confidence intervals are kind of big. We really can't use it. So we scan through all of these until we find signals that meet our criteria. And that's what we see here. So here we've put some restrictions on the molecule and comparator and the incidence rates. So we're only going to look at situations where the molecule has something and the comparator has nothing, or they both have something, and this relative risk, which is the ratio of incidence rates, is greater than or equal to 2 and has a lower 95% confidence interval greater than 1. So here we could say 8.9. And that is for a neoplasm of uncertain behavior of endocrine glands in the nervous system. So this could be a potential safety signal. 4.4, benign neoplasm of lip, oral cavity, and pharynx. Benign neoplasm is like a benign tumor or a polyp or something like that. Ill-defined intestinal infections. Now we actually have a scoring system to use to look at potential safety signals. So if we wanted to do a further analysis, we could say, okay, look, everybody with 237, the patient numbers are low in this particular case, but sometimes they're not. We could do a separate study and just look for that. In addition to ICD-9 coding, soon there will be ICD-10 coding, and we have our CCS. Our CCS is our clinical classification system, which is basically a different form of ICD-9, and we could play the same game with these. Next step, and I'm going to go through these real quick, I think I have 2 or 3 slides left, is treatment pathways. We use these to identify populations, differentiate populations, look at lengths of time on therapy, and investigate possible signals. This is important because understanding these treatment pathways, and this is a very high-level example, if we understand these treatment pathways, we could also understand the length of time on these therapies and also what's going on behind the scenes, right? Are they having any adverse events? Are they taking any other medications? What's going on? And then finally, signal detection for pragmatic clinical trial logistics and planning. So the HIPAA laws that are in place now protect patients' privacy. So when looking at this big data, we get a location on these patients. Some have gone to the state level. but some are still in the county level, and some we could even get the first 3 digits of the zip code. So doing that, not only can we see what the signals are, but we could see where the signals are. So if there are any signals that are worth investigating, especially if we have to set up a pragmatic clinical trial, using something like this, we could know where they are. And oftentimes we can both find what the signals are and where the signals are using the same database. So after we find out what these signals might be, we could look a little bit deeper into the patient characteristics, we could look a little bit deeper into logistics, and then we can make a case to examine further. And that's all we got, guys. Thank you for coming, and thank you for listening. I appreciate it.
Frank, I have a question. How much, or in today's environment, where you have patient-reported outcomes, patients like me, whatever the case may be, where people are part of these social networks, exchanging information, etc. How much does that impact the kind of analysis that you're looking to do?
When you say a patient-reported outcome, do you mean a patient-reported outcome of a standardized instrument?
In some cases, it might be.
Or
There
just-
might be patients that are diabetics, or you see it more with disease registries where, I'm suffering from multiple sclerosis, and, there are no cures. These are the therapies that are out there. People are actually providing information about, how particular therapies are working for them. Does that, is that factoring into the kinds of analyses that you're looking to do?
You're just saying in non-governed social networks, just people explaining how they're doing. It doesn't, but it could. Not directly to what we do using the big data, but perhaps to help develop PRO instruments for these patients and ask the right kind of questions based on what we're hearing from them in these social networks. We don't do a lot of that directly, develop instruments, but we do use the data that comes in from them.
Could you tell us a little bit about your company and the industry that you're in? Because I think a number of our students might be interested in.
Sure. Well, what we do is really 3 things. We're in 3 main areas of healthcare. The first is health economics and outcomes research. That sometimes uses big data, and actually most of the time it does. Because what health economics and outcomes research is really what it says. The economics of a new drug coming to market, which is very important for pharmaceutical companies. What can they charge? Who should we compete against? What line of therapy should we go to? And other health outcomes to consider. To help our clients and companies with these decisions, we put together models, health economic models, budget impact models, revenue impact models, safety models, to help elucidate how our therapy compares to the current standards of care. These are mostly done in Microsoft Excel. So it's like, wow, that's good. I've been using it forever. And we make it pretty with VBA overlays and sometimes Python overlays for some data visualization. We like to do a lot of our data visualization in-house and make it customizable. So a typical project for health economics and outcomes research could start with a systematic literature review. We'd learn about the indication, understand the target product profile and its value statement and where it wants to go, and then build a model around it and look at different scenarios and look at different subgroups. So there's a lot to it. The second aspect is more data analytics and epidemiology, and that's really programming. And it's getting into these databases, writing your own algorithms, understanding the algorithms, extracting the right patient populations, doing the right statistical analysis, and showing what this data can do for these indications. And then the third part is scientific communications. How do we explain the results? And part of it is slide decks, manuscripts, posters, abstracts, and other tools that we build for our clients. Sometimes we take a small subset of data, put a nice interface or an overlay on it, and give it to them as a tool that they could look at different characteristics in different populations within a certain indication or therapeutic area. So those are really the three main things that we do. The industry that we're competing in is pretty crowded, right? Our clients are pharmaceutical companies, biotech companies, but I know a lot of you see also how this could be applicable to finance as well. Because if we're looking at a micro-cap pharma stock that has 1 or 2 products with, dates coming up for their clinical trials and reporting of results, we can look at the preliminary results. We could look at the current standards of care. We could look at what's working on the market and where we expect our drug to be and say this drug is a buy, this stock is a buy, or this stock is a sell. We work in a pretty crowded market, but we're a small company, and I think that gives us advantages. A lot of the companies that do what we do are big, publicly traded consulting companies in the industry. And they're very well known. But what we bring to the table is client service that's personalized to a level that they can't get with bigger companies. And also database expertise. And less red tape. So with us, they have an interface directly to our back office. And with a lot of the big firms, which are great, a lot of times what we hear is the people who started our project are different than the people who finished our project and what happened in between. So it's one of the advantages we have, but it is a crowded space, and our best sales tool, I believe, is our previous work that we do for clients. And one thing that I think separates us, and hopefully was apparent in the slides, was our communication piece. We try and make things easy to understand and visually aesthetic, so that if we do a project for a client, it can be shown in the boardroom to VPs or analyzed in the back office by analysts. And it'll have that scalability where we could look at the top line results and drill down all the way into the details, keeping everything transparent with no black boxes.
Thank you very much.